Here's my chicken leg sample, my workstation, and the tools I'll be using. The first step in the project was collecting the cartilage. I began by carefully dissecting the chicken leg to expose the knee joint. Using a scalpel and forceps, I separated the muscle and tendons to locate the articular cartilage, which is a smooth white tissue covering the ends of the bones where they meet in the joint. This cartilage is responsible for reducing friction and allowing smooth movement. It's also notoriously slow to heal because it has no blood supply. This was definitely a challenging task, seeing as I am not a licensed chicken surgeon and I have never performed such a complex dissection before. Despite that, after isolating cartilage from the knee joint, I attempted to cut it into small uniform pieces, about 5 millimeters in diameter. These samples were immediately rinsed in a phosphate buffered saline, or PBS, which keeps the tissue pH balanced and helps remove loose cells or blood. Next, I dipped each sample into 70% ethanol for a brief surface sterilization, and followed with another PBS rinse to wash away any residual alcohol. This helped prepare the samples for a sterile culture. What I'm currently showing is the end of the chicken tibia. The white tissue covering is the hyaline cartilage. The flappy bit above it is the tendon that holds the patella in place. It wasn't very clear in that shot, but under the tendon is the meniscus, which is a very important part of the knee, made up of fibrocartilage, which is tough and flexible. The meniscus is often injured and damaged in athletes, which isn't the best thing in the world, seeing as the meniscus acts as a cushion and a shock absorber in the knee joint, helping to distribute weight and stabilize the joint. It's kind of hard to run when your bones are grinding against each other. But luckily, I don't think this chicken was an athlete. Here's a better shot of the joint coming up. 1, 2, pause. Here's hyaline cartilage, smooth for gliding across. Play. Pause. Here's fibrocartilage, tough and flexible, used for cushion. Play. I'm trying to extract it fully to use in my experiment. However, that's a little tough to do, seeing as it's embedded within the tissue. And here is where I'm able to get it, and if I can just freeze. There we go, the chicken meniscus. We got it. Play. Day 2. Next, I moved on to preparing the gene delivery system. Here's my workstation, the containers for my treatment and control solutions, my lipofectamine, ampicillin, and TGF-B1, and the pipettes I'll be using. The goal is to get the cartilage cells to start producing more healing proteins, specifically TGF-B1, or transforming growth factor beta-1. This molecule plays a central role in stimulating the production of cartilage matrix, especially proteoglycans and type 2 collagen, which helps give cartilage its strength and shock-absorbing properties. To deliver the TGF-B1 gene, I used a plasmid, a circular piece of DNA with the TGF-B1 gene attached under a strong promoter to help express the gene efficiently. But cells don't just absorb DNA on their own. That's where lipofectamine 2000 comes in. Lipofectamine is a lipid-based vector. It wraps around the plasmid DNA, forming tiny spheres called lipoplexes, which merge within cell membranes and help deliver the DNA inside. I mixed the plasmid and lipofectamine in a low serum solution optimized for transfection, and then added antibiotics to keep the culture sterile. After a 15-minute incubation to let the complexes form, I added the solution directly to each disc containing a cartilage sample. Both treated and controlled cartilage discs were cultured in saline. The treated group received the TGF-B1 plasmid and lipofectamine complex, while the control group only received saline. All plates were placed in an incubator at 37 degrees Celsius to mimic body-like conditions. Here I am putting the dishes in the oven without spilling any of the solution, which if I hypothetically did, I would replace the liquid. Cultures were maintained for 10 days with media changes every 2 to 3 days to replenish the nutrients and remove waste. Though I didn't capture footage of the media changes, they were done consistently throughout the experiment. Day 3 to 10. These pictures were taken on day 3 of incubation. At this point, there were subtle changes in the appearance of the treated cartilage. It was slightly more cloudy and opaque, possibly indicating early extracellular matrix buildup. The matrix includes things like proteoglycans, which are larger molecules made up of glycosaminoglycans, long sugar-rich chains that attract water and help the cartilage resist compression. Day 11. My workstation, staining dye, and rinsing water. To visualize the changes in the cartilage matrix, I stained the tissue using methylene blue and eosin. Methylene blue binds the proteins like sulfated glycosaminoglycans, which are key components of proteoglycans. This tint gives a blue color whenever there are high levels of those matrix molecules. After staining with methylene blue, I added eosin, a red counterstain that highlights cell structures and helps contrast the matrix. The full process involved thinly slicing the samples to see through the tissue structure, then adding drops of methylene blue to sit for 10 minutes. After rinsing, I added the eosin, briefly incubated, and then washed the slides before mounting them for observation under the microscope. These images show the first round of stained cartilage sections. Since my initial slices were too thick, some areas came out very dark and uneven, making analysis difficult. However, I was still able to get one clear control image that showed how the cartilage looked without the TGF-B1 treatment. Day 12, kinda. Here's my workstation. To improve the image clarity, I used paraffin embedding to make thinner, cleaner slices. I began by dehydrating the tissue, soaking each piece in a series of ethanol water solutions, first 70%, then 80%, and finally 90% ethanol, each for 2 minutes. I used a pipette to carefully replace the solutions between steps. Once dehydrated, I poured warm liquid paraffin wax over the samples, fully submerging them and allowing the wax to solidify. This immobilized the tissue, allowing me to cut thinner than I previously could. These thinner sections absorb the stains more evenly, giving me a clear image for comparison. Here are the final stain sections. Compared to the control, the treated samples show a deeper blue coloration, meaning more glycosaminoglycans, and therefore more protoglycan production. This supports the idea that TGF-B1 gene delivered helped regenerate parts of the cartilage matrix, which is a promising result for future cartilage repair methods.